Okay, so welcome back everyone. I think we can get started. So this week we're talking about vision, and this will begin to relate some of the concepts that we've learnt about in previous weeks about the biology of neurons to some everyday features of our experience and our behaviour. To introduce the topic, I thought I'll start with this image that I think many of you will recognise. So it's an image of the dress that went viral on social media in 2015. And to illustrate what's interesting about the image, we can do a quick poll of the class. Okay? And so the question here is very simple. It's just, what colour is the dress? Is it blue and black? Is it white and gold? Or is it some other combination of colours? Okay, so it's not a trick question, it's just reporting the colour of the dress. And you can argue with the person next to you if they disagree. Okay, so you can see the results of the survey in the app, and you can see at the moment that it's about 2/3 of the class are saying that it's blue and black, and about 1/3 that it's white and gold. And these are the results of a similar survey in a larger sample, where again you can see it's a similar pattern of results. More than a half are saying it's blue and black, about 1/3 that it's white and gold. And so there's this strong polarization that occurs in how people see the colour of this image. And it's worth noting that this doesn't just appear to be a matter of semantics. It's not that people are using different words to label the colours that they see. It does appear to be a genuine difference in how people are seeing the colour of the image. A basic principle of colour vision is that the colour that we see when we look at an object depends on the wavelength of light that's entering our eye. And so Isaac Newton famously demonstrated that sunlight can be split into all of the colours of the rainbow, and he did that by passing a beam of sunlight through a prism that separated out the different wavelengths of light that it contained. Light is often described on a spectrum like what you see here, where shorter wavelengths are represented as blue and longer wavelengths of light as red. But an image like the dress poses a problem for this understanding of colour, because when we look at this image, it's the exact same wavelengths of light that are entering my eye and entering your eye, and yet we may see completely different colours. So in this lecture, I'm going to give you a general overview of what the process of perception involves. I'm then going to focus for a while on the retina and talk about how our retina detects light that enters our eye. And I think the interesting part here is how we can use what we know about the biology of the cells in our retina to explain many features of what our visual experience is like in day-to-day life. We'll then talk about colour, and so I'll introduce you to something called the trichromatic theory of colour vision, and then I'll talk about how the colour that we see also depends on the context that we're in. And so this will go some way towards explaining how individual differences in colour perception occur, like the phenomenon of the dress. By the end of the lecture, you should be able to explain how light is detected in the retina, contrast the property of rod and cone cells, which are these cells in our retina that detect light, describe the trichromatic theory of colour vision, and explain why colour perception depends on the context that we're in. Okay, so I can start by trying to define what perception is. And so perception, what I mean by this is our experience of the environment around us that's provided by our senses. And so it's what we see, what we hear, what we taste, what we feel, and so on. So at the moment, as you look around the room, you see your classmates, you see me, you hear my voice, you see the chairs and so on, and that's your perception of the environment. An intuitive way of thinking about perception is that we are directly perceiving the world as it is, because this is generally what it feels like. So in the morning, we wake up, we open our eyes, and the world is just there. We don't have to think about what's there. We just see it in front of us. It just appears in front of us. But the world that you see, that what you see in front of you right now, is arguably not the real physical world. Rather, it's a representation or a model of the world that has been generated in your brain. And the model of the world that is being created in your brain is likely to be different from that created in the brain of the person sitting next to you. And so the study of perception is about trying to understand the processes and mechanisms that are working behind the scenes in our brain to produce this experience of the world. The process of perception can be broken down into a number of different steps that are summarized here, and this takes us from the moment that we look at an object to the moment that we have a conscious experience of seeing that object. And here it's illustrated for vision, but the same concepts apply to all of our other senses as well. So we start with some kind of environmental stimulus, some object in the environment, in this case the tree. And for us to see the tree, there first needs to be some kind of information that's transmitted through the environment to our sensory organs, like our eyes. And so in the case of vision, that occurs via light. And so light is generated in the environment, it reflects off the surface of objects around us, and then it bounces into our eye. And light that is reflected off the tree, in this example, forms an image on the back surface of our retina. So a particular pattern of light falling on our retina that carries information about the presence of that object in our environment. And so that pattern of light falling on the retina is the initial input to our visual system. Our retina contains specialized neurons called receptors. And so these are the rod and the cone cells that we'll come back to later in the lecture. And these cells have the property that they can react to light. And so when light falls on our retina, it stimulates an electrochemical response to occur in these cells. And this is a process that we refer to as transduction. It's where information carried in an external stimulus, in this case light, but it could also be sound waves, for example, is being transduced into information that's carried in a pattern of signals inside of our nervous system. So what we end up with are this pattern of electrical responses in the cells of our retina that are carrying information about the presence of the tree in our environment. That initial stimulation of the retina is actually only the first stage of vision. It isn't sufficient in itself to produce an experience of seeing something. Rather, what happens next is that there's this extensive processing of those signals that occurs across a vast network of neurons that extends from our retina and across sort of large parts of our cortex. That processing of signals across a network of neurons somehow generates a conscious experience of seeing the tree. And so that's our perception. We can then place that experience in some kind of semantic context. We can recognize that it's a tree as opposed to a person or some other kind of object. And then that perception and recognition of objects around us forms the basis of all of our behavior, our ability to interact with the world around us. It's also worth highlighting here that what we perceive, what we see, for example, isn't just affected by the pattern of light falling on our retina. It's also influenced by what we can call top-down processes that are operating in our brain. And so this refers to any kind of information or expectations that are stored in our brain and also influence the way that we interpret the sensory signals and influence what we see. So for example, in this case where we're kind of looking at a moth, the way that the moth appears to us will be affected by the way that it reflects light into our eye. Those are the sensory signals, but it will also be influenced by things like expectations about how moths look, about the kind of objects that exist in the world. And so I'll give you some more specific examples of top-down processes in perception later in the lecture. It may also be worth kind of highlighting that visual perception is occurring in our brain, not in our sensory organs. And so in the case of vision, the eyes are necessary to detect light, but they're not sufficient to produce our perception. So we saw in the previous lecture that people who have suffered damage to their brain, to the occipital cortex, for example, can become blind even though their eyes are working perfectly. And so perception is generally thought to be generated through the activity of a network of neurons in our brain. So the key points here are that perception depends on these specialized cells called receptors that are able to convert some kind of external stimulus, like light, into electrical signals in our nervous system. And then those signals are processed by an extensive network of neurons to produce our perception. Okay, so vision depends on light, and light is produced by different sources in our environment. So by the sun, by the lights in the ceiling, the projector, your mobile phone screen, and so on. And for us to see an object, generally what needs to happen is that light needs to reflect or bounce off that object and then enter our eye. Light is a part of the electromagnetic spectrum that includes other things like X-rays, gamma rays, radio waves, and so on. And so these are different forms of electromagnetic energy that can travel through space and differ from each other in terms of their wavelength. So X-rays have a shorter wavelength than visible light. Radio waves have a longer wavelength. And what we refer to as light is just electromagnetic energy that has a wavelength within a particular spectrum of around 400 to 700 nanometers. The light that's produced in our environment, it reflects off objects around us, and then it enters our eye through the pupil, of course. As it enters our eye, it also travels through these other transparent structures called the cornea at the very front of the eye and the lens just behind the pupil. And the cornea and the lens have a role of bending and focusing the light onto our retina to form a sharp, clear image on our retina. The retina itself is this thin layer of cells, mostly neurons, that coats the back surface of our eye. And this is where the rod and the cone cells are found, which we'll talk about in just a moment. And then the retina also has this structure called the optic nerve, illustrated here, which carries signals from the rod and cone cells into the brain. So this is another cross-sectional image of the retina. So here we're focusing on a part of the back surface of the eye, and the pupil and light will be over that side of the image, so light is entering the eye this way. The retina consists of three main layers of cells. So at the very back of the retina, we have the receptors in red here, the rod and the cone cells. Then we have a layer in blue, which is mainly made up of something called bipolar cells, which are also a type of neuron. And then we have this third layer of ganglion cells, another type of neuron, and the axons of those ganglion cells are extending out of the eye through the optic nerve into the brain. So what happens is that light enters our eye, it passes through this layer of cells and stimulates a response in the receptors that are located at the back here. The receptors share a synapse with the bipolar cells and are able to stimulate a response in the bipolar cells. The bipolar cells share a synapse with the ganglion cells, and when the ganglion cells are stimulated, it generates action potentials that travel down the axon of the ganglion cell into the brain. And so the initial input that our brain is getting about the outside world is this pattern of action potentials that are coming down the optic nerve. These photoreceptors, the rod and the cone cells, there's an image of kind of what they look like on the left here, as seen through an electron microscope. And you can see why they're called rod and cone cells. They each have these outer segments that are either cone-shaped or cylindrical. And these outer segments contain these particular chemicals called photopigments, which are chemicals that are able to react to light. At the other end of the cell, we have structures where the photoreceptor forms a synapse with the bipolar cells. These photopigments, so chemicals that react to light, contain two main components. They contain a protein called opsin together with a light-sensitive molecule called retinal. And light that enters our eye and travels through the retina, through the receptor cells, when it collides with retinal absorbs energy from that light and changes shape. And that change in shape of the photopigment allows it then to interact with other chemicals inside of the photoreceptor, and that triggers this cascade of chemical interactions that ultimately leads to ion channels opening or closing in the photoreceptor that alters the voltage of the cell, that affects whether it's releasing neurotransmitter onto the bipolar cells. And so this is the process of transduction, where energy from the outside world, light, is being converted into a kind of electrochemical response inside of our nervous system. The chemical events that happen inside the photoreceptor are illustrated here, and this is a level of detail that goes beyond the scope of the course, so don't worry too much about these details when it comes to the assessments. But what this figure is illustrating is where we've zoomed in on a particular receptor in the retina, a rod cell, and we find within that cell there are these layers of membrane that have these photopigment molecules embedded within them, these opsin molecules. When retinal absorbs light, it changes the shape of the photopigment molecule, and that allows it to interact with other chemicals inside of the cell. And then there's this pathway of different chemical reactions that stimulates that leads to either the production or destruction of particular molecules that are able to bind to ion channels in the membrane of the cell and open or close those ion channels. And then if they're open, it allows ions to move into the cell. That changes the voltage of the cell and affects whether it's releasing neurotransmitter. Okay, so these rod and cone cells both have the job of detecting light that falls on our retina, But they also differ from each other in a number of interesting ways that affect different qualities of our vision. And so one way that they differ is just in how they're distributed across our retina. So our retina can be sort of divided into two main areas. We have a really tiny area called the fovea, which sits at the very back of our retina opposite the pupil. And this is the part of our retina that is responsible for the very center of our visual field. So when we look at something, we look at somebody's face, for example, what we're doing is we're rotating our eye to bring light that's reflecting off that person's face onto our fovea. The fovea is very densely concentrated with cone cells. So there are actually no rod cells in the fovea, just a high concentration of cone cells. And then the rest of our retina we can refer to as being the periphery of the retina. And it's responsible for everything else that we see in our visual field outside of that very central region. The periphery has some cone cells, but not many compared to the fovea. And instead it has a high concentration of rod cells. So one way of illustrating the different contribution of the fovea and the periphery of our retina to vision is by considering different patterns of vision loss, different patterns of blindness that can occur. And so there's a condition called macular degeneration, which can lead to degeneration of cells, which tends to occur around the fovea. And as a result, the person can experience a pattern of blindness that is illustrated on the left here, where the idea here is that the very center of their visual field is blind. So whenever they kind of move their eyes to look at a particular person, they won't be able to see that particular person, but they will be able to see things in their periphery around that central point. There's another condition called retinitis pigmentosa, which also involves degeneration of the cells of the retina, but tends to occur primarily in the periphery of the retina rather than the fovea. And that leads to a pattern of blindness that is sometimes referred to as tunnel vision, where the person can see the very center of their visual field, but they're blind outside of that central region. Another feature of our retina that's kind of interesting and worth mentioning is this kind of natural blind spot that all of us have in our visual field. And this relates to the position of the optic nerve. So we saw before how the optic nerve is formed by the axons of the ganglion cells that are carrying action potentials from the retina into the brain. And the point of our retina where the optic nerve, where those axons are leaving the retina, leaving the eye, is it doesn't have any receptor cells. And as a result, that means that when light from the outside world is falling on that part of the retina, it's not detected and so isn't seen, if you like. And this does actually lead to a blind spot in our vision that you can demonstrate for yourself. And so I think this will probably work better on your own device rather than on the projector if you have it, and if you have your device about arm's length away. And what you should do here is close your right eye, start with your left eye focused on the yellow dot, and then slowly move your left eye, the focus of your left eye from the yellow dot towards the right across the different numbers. And what you should find is that there's a certain point, so for me it happens at about the number 4 or so, where the yellow dot just disappears. Okay, and so what's happening there is that you're rotating your eye until you get to the point where light from the yellow dot is coming at just the right angle into your eye to fall on your optic nerve. And as a result, that light isn't detected and the yellow dot disappears from your vision. And so it raises the question then of why, if we all have this blind spot in our vision, why don't we notice this? Like, why isn't it just constantly annoying us? And the answer to that is partly because we have two eyes. And so with this demonstration, if you find the point where the yellow dot disappears and then you open your right eye as well, then you'll probably find that yellow dot appears again. And so implying that light from the yellow dot, that when it's falling on the blind spot of one eye, is falling on the retina of the other eye. It's not falling on the blind spot of the other eye. So each of our eyes is able to compensate for this blind spot in the other eye. But then, interestingly, another process that helps here is that our brain is actually able to fill in missing information in our vision. And so what you might notice is that when the yellow dot disappears, you probably don't see kind of just a blank patch. Rather, what most people find is that they just see a complete blue square. So it's like the brain has taken information from around the yellow dot and used that to fill in the missing information in the middle. Okay. All right, so one other interesting feature of our vision is that it adapts to the environment that we're currently in. And so we'll see a couple of different examples of this, but one example that we all experience every day is called dark adaptation. And this is where the sensitivity of our vision adapts to the level of light in our environment. And so, for example, if we're home at night, we have the lights on in our living room and it's quite bright, and then we walk from our living room outside to throw out the trash or something like that, or maybe we walk to our bedroom where the lights are not on, then when we first step into that unlit environment, at first it might be that we can't really see much at all of what's around us. But if you then spend a few moments there in that darker environment, over time your vision will gradually adjust and you'll be able to see more clearly in that dimly lit room. So what happens when our visual system adjusts to the level of light in our environment? How can it change to become more sensitive to light? There seem to be a few different processes that underlie this, but one process relates to the concentration of photopigments in our photoreceptors. And so we saw earlier how transduction involves this process where the retinal molecules in these photopigments are absorbing light and then changing shape. What happens is that they also actually break away from the opsin molecule. And when that happens, the photopigment then becomes unresponsive, or sometimes it's referred to as bleached. And so in order for our photoreceptors to continue detecting light over time, they need to be constantly regenerating these photopigments back to their original state, so that in the future, when more light comes in, they can react to that. At any given moment, each photoreceptor in our retina has millions of these photopigments, and some proportion of them will be in their bleached, unresponsive state, and some proportion will be in their sort of normal responsive state. And so when we're in a bright environment, there's a lot of light coming into our eye, which causes a greater proportion of the photopigments to be in their bleached, unresponsive state. When we first step from that bright environment into a dark environment, we still have sort of a relatively low concentration of the responsive photopigments, but then because there's less light coming into our eye in the dark environment, it gives our photoreceptors a chance to catch up and kind of regenerate more of the photopigments back to their responsive state. And as the concentration of responsive photopigments increases, that also makes our photoreceptors more sensitive to lower levels of light. And we can measure that adaptation process happening, so for both the rod and the cone cells independently. And so in this plot, we have, in the x-axis, we have the amount of time in minutes that somebody has spent in a dark environment. And so this adaptation process is happening over the course of 20 minutes or so here. In the y-axis, we have a measure of how sensitive the cells are to light, where lower values here represent greater sensitivity, so an ability to detect lower levels of light. And so both the rod and the cone cells begin adapting straight away and increase their sensitivity over the first couple of minutes that we're in a dark environment. But then the rod cells continue to adapt over time and end up being quite a lot more sensitive to light than the cone cells. And so the result of this is that when we're in a dark environment, like we wake up in the middle of the night and we're walking around our house, it's primarily our rods that are providing our ability to see in that environment. Okay. So we've talked about these differences between rod and cone cells, but there are also different cone cells, different types of cone cells. And in particular, the main way that they differ from each other is in the particular wavelengths of light that they're most sensitive to. And so we can refer to these as being short, medium, and long wavelength cones, respectively, SM and L cones. And these different types of cones in our retina each have slightly different photopigments that just respond to light of a different range of wavelengths. So that's illustrated here. So along the x-axis, we have the wavelength of light that's entering our eye. In the y-axis, we have a measure of how much each of these three types of cone cells will respond to that wavelength of light. So for the S cones, for example, there's actually a range of different wavelengths that they will respond to, but they will respond maximally to these wavelengths in the shorter end of the visible spectrum. And this feature of our cones, that they act as a system where there are these different types of cones that respond differently to different wavelengths of light, is the basis of our ability to see colour, and we'll return to that in a moment. Okay, so the key points here are that cones and rods, these photoreceptors in our retina, convert light to electrical signals in our nervous system. But they also differ from each other. They differ in their distribution across the retina, their sensitivity to low levels of light, and their sensitivity to wavelength. Okay, so are there any questions at this stage? Okay, so one final way that the rod and cone cells differ from each other that's worth talking about is how they're connected together with other cells in our retina, which we can think of as being the wiring of cells in the retina. So we saw before how the fovea is packed with cone cells, has a very high density of cone cells. And in addition to that, there's also this low convergence between the cone cells and the bipolar and ganglion cells. And so what I mean by that is that typically each cone will send an excitatory signal to just one bipolar cell that will forward that signal to just one ganglion cell. And the result, what that means is that when a ganglion cell fires and produces an action potential that's sent to the brain, it's signaling to the brain the activity of something very precise. It's the activity of that one particular cone cell. And this leads to highly detailed vision in the center of our visual field. In the periphery of our retina, in contrast, we saw before how there are some cone cells, but it's primarily rod cells that are present. And in addition, there's this high convergence of the rod cells onto the bipolar and ganglion cells. So each of the ganglion cells will be effectively collecting signals from a big or a number of rod cells that are all located next to each other on the retina. And so when that ganglion cell becomes active and sends a signal to the brain, it's sending information to the brain about the activity of a kind of larger patch of our retina. The result of that is that we have less detailed vision in the periphery of our visual field, so lower spatial resolution. But it comes with the benefit as well of greater sensitivity to faint light. So to explain this trade-off between the detail of vision and sensitivity to light, we can consider a simple wiring diagram that looks like this. And so what this is representing is light falling on a small patch of our retina. On the left, we have a set of rod cells that are all sending excitatory signals to a particular ganglion cell. And so there are also bipolar cells here, but they're not represented in the figure. And we have, in the case of rod cells, we have this convergence of signals onto ganglion cells. And so this leads to spatial summation, like what we talked about a couple of weeks ago, where if we imagine, first of all, that the light falling on the retina is only a faint light, it might only cause a weak response in each of these photoreceptors, which here is labeled as a response of 2. But because all of those weak responses have been sent to the same ganglion cell, together the ganglion cell is receiving much stronger stimulation. And as a result, if that stimulation exceeds the threshold required to produce an action potential, then the ganglion cell will send a signal to the brain about the presence of that faint light on the retina. In contrast, in the fovea, we have the cone cells, represented on the right here, that are each connecting one-to-one with ganglion cells. And so again, if we have faint light falling on our retina that's producing only a weak response of 2 in each of the cone cells, each of those ganglion cells is only receiving weak stimulation, and that may not be enough to trigger an action potential, and so there's no signal being sent to the brain. So the brain doesn't detect any light on the retina. So the spatial summation of signals, this convergence of rod cells onto ganglion cells, increases the sensitivity of the ganglion cells to faint light. The downside of this convergence of signals is that it reduces the level of detail that's being sent to the brain. And so again, on the left here, we have the rod cells converging their signals onto a single ganglion cell in red. And now we're going to compare two situations where the spatial detail of light differs very slightly. So on the left, we have these two spots of light that are landing right next to each other on the retina, stimulating two adjacent rod cells. In the next scenario, we have, again, two spots of light that are landing on slightly further apart on the retina and stimulating two different rod cells. In both of these situations, it will stimulate an action potential in this one ganglion cell. And so from the perspective of the brain, it has no way of discriminating these two situations. All it knows is that this ganglion cell is active, so there must be light somewhere amongst that cluster of rod cells, but it doesn't know exactly where. In contrast, when we have cells like cone cells connected one-to-one with the ganglion cells, in these two situations where we have spots of light landing on slightly different parts of the retina, as long as the light is sufficiently bright, it will stimulate different ganglion cells, and as a result, there will be a different pattern of activity being sent to the brain in these two situations, and so the brain is able to discriminate that slight difference in where light is falling on the retina. And so this is a significant part of what underlies the difference in the clarity of our vision between the center of our visual field and the periphery. And so generally things look kind of blurry to us in the periphery. Like, for example, when we're reading a book, we don't just kind of stare in the center of the page as we read the book. Rather, we're constantly moving our eyes from word to word. And that has the effect of bringing light from each word onto our fovea, where we're able to detect enough spatial detail to read the text. And you can sort of demonstrate that kind of idea to yourself pretty easily here if you keep your eyes fixed on the cross on the right. Then I reveal some letters to the side. Probably you find it difficult to read those letters when they're in the periphery. Okay, so the key points here are that cones in our retina are connected up to ganglion cells in a way that promotes better visual acuity or spatial detail in our vision, while the rods are connected up in a way that promotes visual sensitivity at the expense of acuity. This table is from the textbook and just summarizes some of the differences between rods and cones and the fovea and the periphery that we've talked about. Another way of summing it up is with an image like this, where the idea here is that the kind of vision that our cones give us is represented at the top here. So they give us a sense of color, fine spatial detail, so we can sort of read the words on the spine of the books, and they operate in bright environments, whereas at the bottom we have a representation that's more like what rods give us in terms of vision. So things look more blurry, they lack color, but rod vision operates in dim environments. And so if we wake up in the middle of the night and we look at our bookcase, it's more likely that it will look like what you see at the bottom there than at the top. Okay, so we can have a break in a moment, but before we do, let's do a quick poll everywhere question. So the question here is about ganglion cells, and so ganglion cells in the retina that receive signals from a small number of photoreceptors will tend to provide which of the following? Greater sensitivity to faint light, worse discrimination of color, more detailed vision, or less detailed vision. So I'll give you a moment to put in a response. Okay, so most of the class put C, which I agree is the right answer. So we've just been talking about this effect of convergence of neural signals onto ganglion cells and how that affects the clarity of vision, the level of detail that our vision has. And where kind of counterintuitively, in a way, a smaller number of cells sending photoreceptors, that is, sending signals to a ganglion cell, actually results in overall in better spatial detail, because it generally means that the signals are coming from a more precise area on the retina compared to if a larger number of cells were all sending signals to the ganglion cell. So that's why C is correct rather than D. And then it has this trade-off, however, that with fewer cells sending signals to the ganglion cell, it can lead to less sensitivity to faint light, because you have less of that spatial convergence of signals that can allow a ganglion cell to pick up very faint levels of light. And in terms of color perception, our ability to discriminate between different colors relates to the sensitivity of cells to the wavelength of light, and we'll talk more about that in the second half of the lecture. Okay, so let's have a break just for 5 minutes or so and come back at 5 minutes to 3. Thanks. Okay, so I should get started again. So for the rest of the lecture, we're going to focus on colour vision. And our perception of colour, as we've mentioned, relates to the wavelength of light that's entering our eye. And so when we look at an object, there's generally light reflecting or bouncing off the object into our eye. And importantly, the way that light reflects off an object differs between different objects depending on the wavelength of the light. So there's a process called selective reflection where the light falling on the object, some of the wavelengths will be absorbed by the surface of the object and other wavelengths will be reflected by the object. And it's those reflected wavelengths of light that enter our eye and determine how we perceive the object. Okay, and the particular wavelengths of light that an object reflects depends on the material of the object. And so I think a nice example is a ripening tomato. That an unripe tomato is rich in a compound called chlorophyll, which tends to reflect more medium wavelengths of light. And as a result, we tend to see an unripe tomato as being green. As the tomato ripens, there are chemical changes that happen in the tomato. Chlorophyll is broken down, other compounds are produced, and the result is that the tomato starts to reflect more longer wavelengths of light. And now we see it as red. There's a phenomenon that's really useful for understanding how our perception of colour operates, which is called additive colour mixing, and that's described in this video.
There's an antique demonstration that goes back long into the 18th century, but which still has a great magic when one sees it for the first time, in which one mixes a red and a green and a blue light. One begins with a red patch of light, and the most remarkable result is that of mixing with that red a green light. One sees a very vivid yellow where the two overlap. And if now one adds, thirdly, a blue light, where the three overlap, one has a very plausible white. Where the green and the blue overlap alone, then one has cyan, and where the red and the blue overlap, one has a purple. This demonstration illustrates the most fundamental fact, perhaps, about human colour vision, the fact that you can produce all colours by mixing three. And this fact was known quite early in the 18th century, but it was miscategorised. It was thought to be a property of the world, a property of physics, rather than a property of people.
Okay, so just to recap a part of what he's describing there, he's talking about this situation where we start with a flashlight that produces a longer wavelength of light. We shine that on the wall, some of that light reflects into our eye, and we see the colour red. We then take a flashlight that produces a more medium wavelength of light, and in isolation we would see that light as being green. But when we shine the flashlight on the wall together with the longer wavelength flashlight, so now we have both longer and medium wavelengths of light entering our eye when we look at the wall, we now don't see red or green, we see a unique colour, which is yellow. This process of additive colour mixing, where we can mix together different wavelengths to create novel colours, is the basis for almost all of our display technologies. And so your computer screen, your mobile phone, the projector and so on all work on this basis. And so your mobile phone screen, for example, is made up of a grid of pixels. And if you zoom in on the screen you would see something like this, where each of those pixels is made up of 3 different sub-pixels, or light guns, they're also called, where one of those sub-pixels is producing blue light, another is producing green, and the other is producing red light. And by varying the intensity of that, the red, green and blue light that is produced by each pixel, the screen is able to create the impression of any colour. So additive colour mixing describes this process where there are these 3 what we call primary colours, red, blue and green, that we can mix together in different quantities to produce the perception of any other colour. So if we have red, blue and green light all shined into our eye together, we would see white. If we have just green and red together, we would see yellow. And then if we modulate the intensity of the two, if we still have red and green, but we sort of dial down the green a bit, then we'll probably see something like orange instead. And this brings us to the trichromatic theory of colour vision. So we saw earlier how there are these 3 types of cone cells in our retina, the S, the M and the L cones. And these are each sensitive to a range of to light in a range of different wavelengths, but they differ from each other in terms of the wavelengths that they respond best to. These 3 types of cones are just intermingled across the surface of our retina, and particularly in the fovea. And so on the left here we have an image of a real person's fovea, or a small part of it, where they've identified, so these are individual cone cells that you're seeing here, and they've identified whether these are S, M or L cones, and then used these false colours to label the different cone types. So you can see that these 3 types of cones are just kind of intermingled across the surface of our fovea. And these 3 types of cones that respond to light differently depending on the wavelength shares this obvious parallel with the phenomenon of additive colour mixing, where we can use 3 primary colours, red, blue and green, to create the impression of any other colour. So the idea with the trichromatic theory of colour vision is that the colour that we see when we look at an object depends on how light that's reflecting off that object is stimulating these 3 different types of cone cells. And in particular, that each colour experience is associated with its own unique pattern of activation across these 3 types of cones. So we see blue when there's stronger activation of the S cones in our retina compared to the M or the L cones. We see green when there's strongest activation of the M cones compared to the S cones or the L cones. And we see red when there's strongest activation of the L cones compared to the other 2 types of cones. So the idea here is that colour perception depends on the pattern of activity or the relative response of these 3 different types of cones. And then other colours outside of the primary colours will also be associated with their own patterns of activity across these same 3 types of cones. So we will see yellow when there's strong activation of both the M and the L cones, but weaker activation of the S cone. And we'll see white when all 3 of the cones, types of cones, are stimulated to an equal extent. And so now this gives us an explanation for why additive colour mixing works in the way that it does. And I think this is quite nicely explained in this video.
This lemon looks yellow to me, and it probably looks yellow to you as well, but not in the same way. You see, here in this room, this lemon is subtractively yellow. It absorbs all visible wavelengths of light except for yellow light, which it reflects onto my retina. But the screen that you are using to watch this video doesn't produce yellow light at all. In fact, it can only produce red, blue, or green light. The really cool but kind of disturbing thing about this is that here in the room, I am actually seeing real yellow light. But you are seeing fake yellow. Absolutely no yellow is coming off of your screen and falling on your retina. But it still looks yellow because it's quite easy to lie to the brain. Our retinas contain 3 different types of cone cells that are receptive to color, and each one is best suited to detect a certain color. One is great for blue, the other is great for green, and the third is great for red. Notice that there's no individual cell looking for yellow. So the way we actually see yellow happens like this. The wavelength of yellow light falls between the wavelengths of red and green. And so when an object reflects yellow light onto your retina, both the green and the red cones are slightly activated, which your brain notices and says, well, that's what happens when something's yellow, so it must be yellow. All a computer monitor or a mobile phone screen has to do to make you think you're seeing yellow is send a little bit of red and a little bit of green light at you. As long as the pixels and the little sub-pixels on them are small enough that you can't distinguish them individually, your brain will just say, well, I'm receiving some red and some green. That's what yellow things do. It must be yellow. Even though it actually is not.
Okay. So just to kind of recap the basic idea there, the idea with the trichromatic theory is that our brain, our visual system, is capturing information about the wavelength of light entering our eye by comparing the response that it produces across these 3 types of cone cells that are tuned to more shorter, medium, or longer wavelengths of light. And because of that, it's possible for these errors or kind of miscategorizations to occur where physically different patterns of light can be perceived as having the same color. And so the example used in that video was where if we have yellow light, so meaning light of a particular wavelength of around sort of 580 nanometers or something like that, it will cause stimulation of the L cones. It will also stimulate the M cones, but it won't tend to stimulate the S cones very much. And the important point is we can produce that exact same pattern of activity across the 3 types of cones using other types of light. For example, the combination of a green light and a red light together will also cause stimulation of the L cones, stimulation of the M cones, and weaker stimulation of the S cones. And so because our brain, all it has access to is that pattern of responses across the 3 types of cones, it's unable to distinguish those 2 situations. And so we see them both as being the same. And it's worth maybe emphasizing that this is a property of our biology. It's not a property of physics. So there isn't any physical reason why a yellow light should be treated the same as a combination of red and green. It's just a feature of how our brain and our visual system is representing color or capturing information about the wavelength of light entering our eye. And it's sort of tempting with this theory to think of these 3 types of cones as being each responsible for a different type of light, like that our S cone is responsible for the perception of blue, for example, our L cone is responsible for our perception of red. But something to emphasize here is that in order to capture information about wavelength, our brain needs to compare the pattern of response across all 3 types of cones. It can't just look at one cone individually. And to illustrate why that's the case, it's actually because the response of a single cone type, an S cone for example, is not only affected by the wavelength of light that's hitting the cone, but it's also affected by the intensity of that light. So as an example, if we first imagine one situation where we have a kind of bluish light entering our eye, it causes strongest stimulation of the S cone. It also causes some stimulation of the M cone and weaker stimulation of the L cone. Now consider a kind of red light entering our eye. It causes stronger stimulation of the L cone, but also causes some stimulation of the M cone and less of the S cone. And then if we have a green light entering our eye, it will cause the stronger stimulation of the M cone compared to the other two cone types. But the response that it produces in the M cone also depends on the intensity of light. So if we imagine that that's a weak green light, like we've sort of dialed down the strength of the flashlight, it's running out of batteries, something like that, then it might still only cause a sort of moderate response in the M cone. And so the idea here is that in those three scenarios, we might be ending up with actually the exact same response happening in the M cone, even though the color of the light is completely different. And what that means is that our brain can't tell whether green light is entering our eye just by looking at the M cone. It has to always compare across the three cone types to see which is most active compared to the other two. So that's why the pattern of responses across cones is what's important to capture information about wavelength. Okay, so the key points here are that this trichromatic theory is that our perception of colour depends on the response of three receptors in our eye that are each tuned to a different spectrum of wavelengths of visible light. And in particular that it's the relative response across these three receptors that captures information about the wavelength of light and determines the colour that we see. Okay, so we can again do a quick poll everywhere question to sort of see how you're following along. And so I'll just activate it. All right, so the question here is, in which of the following situations are the M cones and the L cones in your retina least likely to be active? A, when you're looking at a combination of red and green light. B, when looking at a yellow object like a banana. C, when looking at a white object like the sun. Or D, when you're navigating a dimly lit environment. So I'll give you a moment to put in a response, and you're of course welcome to chat to the person next to you about it as well. Okay, so there's a pretty unanimous response. Maybe my questions are too easy. So D is the right answer. So the idea here is that in a dimly lit environment, we have less light entering our eye, stimulating our receptors, and we also saw that in dimly lit environments, it's primarily the rods that are sensitive enough to detect light rather than the cones. So D is a good answer here. And these other three options are really they're all describing situations where the light is different, but still could produce strong activation of the M cones and the L cones. So red and green light, the red light could activate the L cones to a greater extent, the green light could activate the M cones. Yellow light in this part of the spectrum again can activate both of these cones strongly, and a white object, what we call the white light, is light that contains a broad spectrum of different wavelengths that again would tend to activate all of our cones. Okay, so are there any questions about the trichromatic theory? Okay, so to finish up with, we can sort of go beyond the trichromatic theory now and talk about how perception of colour doesn't just depend on the wavelength of light entering our eye, but also depends on the context that we're in. And so we saw this example at the beginning of the lecture of the dress, this image that different people in the classroom here see as having different colours. And we noted that what's kind of profound about this or interesting about this is that it's the exact same information coming into our visual system, the same wavelengths of light entering our eye when we look at this image, but some of us are seeing different colours to others. And our perception of colour, it doesn't just depend on this relationship to wavelength, it also depends on the individual and the context that we're in. So a concept that's, I think, useful for explaining why this is the case is called colour constancy. And this refers to how the perceived colour of objects around us can be relatively stable when we see them across different viewing conditions, despite the fact that the wavelengths of light that enter our eye when we look at that object can differ across different conditions. So there's an illustration of that here, where we have the same set of objects lit under two different sources of light. On the left, they're lit under kind of a pinkish light. On the right, a greenish light. And this is just a feature of our everyday vision, that we're constantly moving from different environments, between different environments that have different types of light sources, like a more bluish lights, more yellowish lamps, the light of the sun, and so on, all contain different types of wavelengths. And that, to some extent, changes the appearance of objects. And so here, comparing across the two images, I would say that the orange is kind of more neon shade of orange on the left compared to the right. The strawberry is sort of a darker looking strawberry on the right compared to the left. But generally, the colours are pretty similar. I'll still tell you that the orange is orange in both images, the strawberry is red, the banana is yellow, and the text on the sort of can of moisturiser is white. And the reason that's kind of interesting and surprising in a sense is that the wavelengths of light that are entering our eye when we look at these objects are actually quite different across these two images. So if we take that white text, or at least I see it as being white in both images, and we isolate the pixels that make up that text and look at them outside of the context of the image, what we find is that the white text in the image on the left is actually pink, whereas in the image on the right it's actually green. So it's not actually white in either of the images, and yet when we see those pixels in the context of the image, they appear white. And the reason for that, the reason that the wavelength of light that they're sort of reflecting into our eye is different, is because of the light source. It's because in one environment even though the object itself, the text is white, there's pink light falling on that text, and so it's reflecting pink light into our eye. And then in the other environment, that same text is reflecting a different type of light into our eye due to a difference in the light source. And so for us to perceive that text as being white, what it implies is that at some level our visual system has been able to separate out the colour of the light source from the colour of the objects. It's somehow able to take that pink light and figure out that that pinkness is due to the light source. It's not due to the object itself. The object is white. And this gives us a kind of explanation for why this dress phenomenon occurs. So at least one type of explanation. And the explanation is that whether we see this image as being a blue and black dress or a white and gold dress depends on whether our visual system at some level is attributing the blueness in the image, the shorter wavelengths of light, as being caused by the material of the dress itself, or being caused by the light source that the dress is illuminated under. And so this image on the right, what it's trying to illustrate is that this exact same image, same pattern of wavelengths entering our eye, could in theory be caused by different things in the world. It could be caused by a blue and black dress that's illuminated under a more yellowish light, or the exact same image could be caused by a white and gold dress that's illuminated under a more bluish light. And so we can, the reason this image is polarizing then is likely because it lacks cues to the illumination of the dress. And so if you're somebody that sees that the dress has been white and gold, what it implies is that your brain at some level is kind of assuming that this dress is lit under a bluish light rather than a yellowish light. But we can try to add in those cues and sort of push our perception in one direction or the other. And so here the dress itself is identical between the two images. It's made up of the identical pixels. But what we've done is just add in this context where we've changed the appearance of the woman's skin to make it appear like she's lit under a more kind of yellowish light on the left or a more bluish light on the right. And this demonstration doesn't work for everyone, but it works really well for me, so hopefully it works for some of you as well. But for me at least, it's clear that on the left she's wearing a blue and black dress, on the right she's wearing a white and gold dress. The two dresses look completely different to me in their colour, even though I know that the pixels in the image are identical. So the idea here is that giving contextual cues to the illumination conditions can affect the way that our visual system is kind of decomposing the wavelength into our eye into those that are caused by the object and affecting our perception of the colour of the object versus wavelengths that are caused by the lighting conditions. Okay, and this illustrates a problem that our visual system is faced with all the time, which is that the light that is entering our eye is actually ambiguous regarding the colour of objects. And so it's ambiguous because the wavelength of light entering our eye can be affected both by the properties of the objects themselves, like the material of the dress or whether a tomato is ripe or unripe, but they can also be affected by the illumination conditions that the object is in. And the reason it's a problem is because generally we use colour to understand the nature of objects around us. We want to, if we go to the supermarket, we want to know whether these tomatoes are ripe or unripe. We don't really care whether they're lit under sort of a more greenish light or a more kind of reddish light, for example. And so our visual system is always making kind of inferences. It's trying to work out to what extent the wavelength of light entering our eye should be attributed to the nature of the object that we're looking at versus the nature of the light source that it's under. It seems to do that partly by drawing on contextual cues to the lighting conditions, as we've just talked about, but also through other processes like adapting to the dominant colour in our environment and drawing on expectations or past experiences that we have of different kind of familiar objects. So to illustrate those two processes as well, we can start with adaptation. And so we talked about adaptation to the level of light in our environment earlier on, but adaptation also occurs to colour as well. So our visual system adapts to the kind of colours that we're exposed to. And we can demonstrate that process of adaptation over the short term quite easily. So if you keep your eyes fixed on the black dot in the center here and keep your eyes still and just fixed on the black dot for 20 seconds or so. And then in a moment, I'm going to change the slide back to a grey background. And when I do that, I want you to still keep your eyes on the black dot, but just notice what you see. Okay. Okay. So what you should see then is that you get these false colours in your vision, these 4 squares of false colour that are actually complementary to the colours that you were previously exposed to. So this short-term exposure to particular colours leads to this kind of illusion of seeing these false colours in your vision. And that reflects this process of adaptation where our visual system is adapting the way that it responds to the wavelength of light depending on the kind of wavelengths that we've recently been exposed to. And we experience this type of kind of short-term adaptation in sort of every day. Like if you kind of look at a street light for a while, then look away, you also get this kind of patch in your vision for a few seconds. And usually they kind of appear just like an artifact of our vision, something that isn't really kind of useful. But it reflects this general process of adaptation that actually does have an important functional role, which is to help normalize our experience of colour across different environments. And so in day-to-day life, we're always moving between different environments where the types of colour, the types of wavelengths of light that we're exposed to differ. So we might go for a walk in the park in the morning and we're in a very lush environment exposed to a lot of kind of green light, like medium wavelength light, and then we come to uni in the afternoon, we're in sort of a drably lit lecture theatre, and the types of light that we're exposed to is quite different. And the idea is that whenever we're in a particular environment, our visual system is adapting to the colours that are present in our environment in a way that tends to make that environment appear more similar to other environments than what it really is. So to expand on what I mean there, if we imagine that we're in this nice lush environment, we're exposed to a lot of green light, so sort of medium wavelength light, the M cones in our retina will be initially responding more compared to the other two cones. They're stimulated more by that medium wavelength light. But as we spend sort of some moments, some minutes or so in that environment, the M cones, because they're stimulated more, will also adapt more. They'll reduce their sensitivity as a result of that stimulation. And that leads to a more even response happening across our different cones in this environment that is effectively filtering out that sort of that dominant colour, that preponderance of medium wavelengths of light. And the result of that is thought to be that environments end up looking kind of more similar to each other than they really are in terms of the colours that are present. And this is relevant to colour constancy because the dominant colours in an environment are often related to the light sources that are present, because those light sources are affecting the colour of everything around us. And so by adapting and kind of reducing our sensitivity to those dominant colours, we're often sort of filtering out the effect of the light source on the colours that we see. Okay, and then one final factor that I'll mention that also contributes to colour constancy and is more generally just an example of how our context affects the colour that we see is the role of expectations. And that can be demonstrated partly with this image here. And so the question here is what colour are the strawberries? And for me at least, I see them as being clearly red, okay? And what's surprising about this is that if we isolate the pixels in the image again, what we find is that there are actually no pixels in the image that individually look red. The pixels making up the strawberries are different shades of grey and white. We can also demonstrate that by covering up part of the context of what we're looking at. And for me, at least, that has a really strong effect of making it apparent now that the surface of the strawberry is not red at all. It's just grey. But once that context is sort of put back into play, I just can't help but see them as being red again. And there are actually a couple of different processes that seem to underlie this sort of illusion of redness, if you like. But one factor that plays a role is our expectations about the colour of familiar objects. And so in day-to-day environments, many of the objects that we see have characteristic colours. So oranges are usually orange, lemons are usually yellow, grapes are usually green or red or something. And so we all have this lifetime of experience of associating particular types of objects with particular types of colour. And there's evidence that this can actually affect the colour that we see when we look at an object independent of the wavelengths of light that are entering our eye. So one study that showed some evidence for this, what they did is they showed images of certain fruits and vegetables on a computer screen, and then they set it up in a way where the subject could press buttons on the keyboard in order to change the colour of the image. So they could make the banana like more blue, for example, or more red, or more green, or more yellow, and so on. And then they asked the subject to make these fruits and vegetables all just perfectly grey, like just make it a grayscale, adjust the colour until it's just perfectly grey. And what they found is that people didn't adjust it until it was actually grey rather they adjusted the colour until it was a kind of opposite colour to the characteristic colour of the object. So in the case of a banana, they adjusted it until the banana was slightly blue, and then they said, this is a grey banana. Okay? And the way that they interpreted this is that when the banana is actually grey on screen, people are still seeing it as being slightly yellow because of these prior expectations about the colour of bananas. And so they have to add this kind of complementary colour, blue, to the image in order to make it actually appear grey to them. And they were able to demonstrate that for a range of different objects that have different types of characteristic colours. Okay, so the key point here is that the perception of colour doesn't only depend on the wavelength of light that's entering our eye. It also can depend on the context that we're in. So cues around us that tell us something about the illumination conditions, for example. It depends on the current state of our visual system, so the kind of colours that we've recently been exposed to and which our visual system has adapted to. And it also seems to depend on things like prior expectations that we have, prior knowledge that we've developed through our life. And so at the beginning I mentioned that perception doesn't only rely on the sensory input, which in the case of colour is the wavelengths of light entering our eye, but it also depends on these top-down processes as well. And so the effect of prior expectations on perception of colour is one example of that. And the concepts that we've talked about here, the idea of receptors, adaptation, prior expectations and so on, are not just relevant to colour, but actually affect all different aspects of our perception. So including perception of speech, for example, perception of other people's emotional expressions and things like that. And so we'll begin to talk about some other aspects of perception in next week's lecture. Okay, so I should finish up. So if you have any questions, then please feel free to come up and ask me. And next week we'll build on today's lecture by talking more about the role of the brain in producing our vision. Okay, thank you. I'll see you next week.